Uh, welcome everyone to the next in the HPC-CONEXS webinar series, and today it's a real pleasure to have Dr. Niri Govind from Pacific Northwestern National Lab, and Niri has done a huge amount of work in looking at electronic structure theory. He's one of the core developers of NWChem. And given that we're talking an X-ray workshop, the core developer was an intended pun. It wasn't an accident. Really, the work in the field has been really important across all types of spectroscopy. But today, what he's going to really talk about is work in explorations in ultrafast dynamics and X-ray spectroscopy. So, as I said to Niri earlier, one of the NWChem, which is the software that he does a lot of work with, is one of the core softwares that's supported by the Archer 2 service. And so this really has direct relevance if you're either interested in some of the techniques and then applying them and using high computing performance computing resources through HPC-CONEXS. So as usual, if you have a question, feel free to put them in the chat as the talk goes along. We can either ask them at the end, or at the end of the presentation, you can give the put your hand up, and yeah. Otherwise, I'll hand over to Niri and look forward to the presentation.
Thanks, Don, and thank you for accommodating me at the early hour at 8 o'clock, and thank you, Sydney, for going back and forth and coordinating everything. Thank you all for attending my seminar. And today, what I'll do is I'll talk to you about some work that we've been doing over the last, I would say, 4 years or so, and quite a bit of it during the pandemic, in fact. And as Tom pointed out, this is all being done with the NWChem code. And I'll show you this in terms of not from a point of view of the implementation but from a point of view of how we've actually used it in creative ways to do interesting science, but more closely with an extra spectroscopy tilt to it. And this picture you see in front here is actually a paper that was published about 2 years ago, in fact, in Nature Chemistry, and we had this nice graphic where we were looking at the ultrafast electron transfer process between an iron and a ruthenium center in a fairly complex transition metal complex sitting in water, actually. And I like this picture a lot. So as Tom was pointing about, just a small short lesson in geography. So this is the state of Washington in the Pacific Northwest, and you might all recognize Seattle right here. Portland is down here. There is this mighty Columbia River that actually flows right at the border of Washington and Oregon. Oregon is down here. And so the Pacific Northwest National Laboratory is actually located in Richland, which is the Tri-Cities area. And if you ever fly into the Tri-Cities area, Pasco will be your airport you'll be flying into. And I like to call this the somewhat an evergreen part of the evergreen state. But we have a lot of blue skies here, in fact, because of we're actually on the leeward side of the Cascade Mountains that run north-south here, in fact. So we're pretty much dry on the east side. All the rain is on the west side here. And these two little insets you see here, this is an aerial shot of our laboratory on the banks of the Columbia River here. And there are a few more extra buildings now here. But this is sort of what it looks like from the air. And this is our esteemed neighbor, in fact, LIGO Hanford, that detects gravitational waves, and it's been in the news lately in the last several years now. So just because the context of this talk is all about X-ray spectroscopies and dynamics, and I'd like to just show you just a brushstroke about where this is relevant, in fact. And all the work that I do is mostly in the context of chemistry, and PNNL, we focus a lot on interfacial chemistry, not so much on the material science side of it, even though there are people working in it. So I'd like to show you just a broad brushstroke in sense of where spectroscopies and dynamics really make sense. And so if you look at the list, they're relevant to ultrafast chemistry because X-rays are these exquisite probes to probe ultrafast dynamics, understand energy flow, elucidate structural dynamics. This can go on to chemistry at complex interfaces, charge transport, and reactivity, reaction pathways, and diverse environments in chemistry and aqueous environments. And these are broad areas that we're actually interested in PNNL as a broad area of science, in fact. So the top here, in fact, and I'd like to point out that last week we did get with the big announcement of the Nobel Prize in Physics being awarded to ultrafast pulses. All of these science areas that I've listed here in the sense basically traverse a whole range of timescales. And I'd like to point out, going with increasing speed going upwards. In fact, if you want to study the trigger mechanisms of a reaction or a fluorescence process, you really have to go down to the very shortest line scales to actually start seeing this. And this is made possible right now with the advent of these X-ray free electron lasers. And here I show you a little snippet of the LCLS facility at SLAC in Stanford, and then this is the Swissfeld facility. And of course, if you go to slower timescales, you do have the synchrotrons that are actually still, I mean, very, very, very relevant for lots of characterizations and still very, very interesting dynamics. And there's also a capability that I don't list here, which is a lab-based capability that's being developed out of Jerry Seidler's group at the University of Washington at Seattle, and who runs the Global XAS webinar series. So with that as the backdrop, I'd like to sketch out a few, in this talk, I'd like to sketch out a few areas that we've been working in over the last several years, and some examples of various applications that we've done with our capabilities that we've developed. And all of the methodology that I'll be using today will be in the context of density functional theory and time-dependent density functional theory. And we all know, experts in the room know, that there are limitations with DFT and TDDFT, but there's a lot you can also do with DFT and TDDFT, as long as you know what you're getting into and basically making.
Understand very much the feedback. For now, I wouldn't put too much effort on the analysis.
Oh, sorry.
Someone was unmuted. Yep. Cool.
All right. Okay. So with that in mind, like Tom pointed out earlier, all of these applications were actually performed with the NWChem code that I'm a member of, and we have this GitHub site that you can actually download the code from. So I'll give you some examples from where we've looked at X-ray absorption spectroscopy from a XANES standpoint, valence-to-core emission, which is the reverse process, de-exciting from the valence to the core, and then how we basically connect up absorption and emission, in fact, to give you RIXS facts. So this audience probably doesn't need an introduction to this, but for the uninitiated, I'll basically go over this very, very quickly. So if you look at an X-ray spectrum over a broad range of, say, about 500 electron volts, this is the shape you normally see. It's like a big S shape with some undulations out here, which is called the XAFS region. That's very well described with multiple scattering techniques. And the FEF code out of the University of Washington does an extremely good job of this thing. And then, of course, as you go to lower energy, you have this XAFS region. And as you get down to where the edge starts to rise, you get these pre-edge regions, which are essentially transitions to bound transitions. And it basically gives you the local chemistry of the system and the coordination and so on. And these have been very, very relevant, in fact, to characterize molecular complexes, dynamics, and so on. And if you zoom into the pre-edge region, right, you can basically X-ray spectroscopies, you can broadly classify them into different edges. The K edge being excitations out of the 1s, the L edges basically excitations out of the 2s and the 2p states, and so on as you go out to lower and lower energies. And people who work with X-ray spectroscopies, especially theory and as well as experiment, know as you go to shallower and shallower edges, the spectrum gets a lot more complex, and it's also harder to model. In fact, you need very precise theory, in fact. So little details matter when you actually go to shallower edges, because you have multiplet effects, spin orbit splittings, and so on, to get a very precise representation of what you're looking at. So everything that I'll show you today basically is within this K and L edge regime, in fact. So the implementation in NWChem, we do have two implementations of time-dependent density functional theory, and I'd like to split them up into what I call linear response, or LR, and a real-time version, which allows you to propagate the density matrix. And I will not be talking about the real-time version in this presentation, but just the linear response version. And similar to other implementations in widely used quantum chemistry codes, we've extended our TDDFT solvers to actually take advantage of excitations out of the core states. And essentially, you basically build a trial space out of excitation, say, if you're interested in the 1s to virtual orbitals, or the 2p or the 2s or 2p orbitals to some virtual orbitals. And once you develop this model space, you can actually solve the TDDFT equations within that model space to get you the excitations you require. Of course, you have to pay attention because these core states do have the benefit that they're actually well separated from the other states energetically. So that allows you to actually define these model spaces really well. If you need further details about this implementation, in fact, we published this paper about over 10 years ago, in fact, that talks about both the real-time as well as the linear response versions of it. So if anybody's interested, you can look at this 2012 paper that was published in JCTC. So getting into the science from the XANES, from the first set of examples, this was a problem that was actually posed to us by a collaborator at the Advanced Photon Source at Argonne. And they had actually were interested in, they had developed some new capability to look at the photoreactivity of aqueous Fe2CN6, which is actually a very good model complex. In fact, if you want to test theory, and people working in X-ray spectroscopy theories know very well that if you have a good implementation, this is a good system to actually go after, in fact, and if you want to dress it up with a good solvation environment and so on. So before I actually get into that problem, this is a paper we wrote in 2018 where we actually characterized the Fe2 system, the 2+ as well as the 3+ system for this low spin because the cyano group is in fact a very, very strong ligand. So we characterized this in this 2018 paper. For all the details, please go and look at this paper where we showed you how this complex can actually be simulated in a full-fledged QMMM environment. And we characterized this, the spectroscopy of the system, of these two systems, all the way from the infrared, all the way over to the X-ray spectroscopies, including valence-to-core X-ray emissions. But here, for the sake of this presentation, I'd like to draw your attention to two key features that show up in the Fe2+ and the Fe3+ complex that will be necessary as we go forward into the next topic that I'll introduce. So in the Fe2+ complex, since your T2G orbitals are actually fully filled, you only have excitations out of the 1s if you're looking at the KH into these EG states, in fact. And what we call them the B features here. On the other hand, if you do an Fe3+ system, you have an open orbital present here in the T2G and two open orbitals present in the EG, and you get the emergence of this A feature here, in fact. So this just a very, very simple metric. You can actually see a simple way of, in fact, seeing what the Fe2+ and Fe3+, and like I was telling you earlier in the XANES, if you look at the pre-edge XANES, it can actually tell you the oxidation state of the element you're interested in looking at. And one thing you can actually would have noticed by now is since you've just basically got one orbital to excite into in the T2G and two orbitals in the EG, you actually see an almost 2-to-1 ratio that you see here, in fact, in the Fe3+ complex between the A and B features. And one thing you should pay attention is since these are 1s to 3D transitions, these are quadrupole transitions. And so when you compute these oscillator strengths, you have to go to higher orders to make sure that you capture all those effects. So let's get into this thing. So the interesting thing that this problem that was posed to us by our collaborators was essentially to study this photoreactivity of aqueous Fe2CN6. And this is well known in the literature. In fact, if you shine light at less than 330 nanometers, and these studies go back all the way to 1971, if you shine light less than 330 nanometers, you basically get photooxidation. And if you go more than 330 nanometers, you get a photoacquation as the dominant mechanism. And what that means is when you shine light, you basically get a rapid excitation. You get a rapid excitation and a quick transfer into a triplet state. And over a course of time, you in fact end up losing one of the cyano-ligands, and one of the waters from the solvent basically comes and fills that position. And that reaction is known to take about 20 picoseconds. And I'd like to point you to good papers, in fact, recently over the last 5 years by the Chergui group that have actually, that gave us kind of a roadmap on how to go about looking at this, but they also left gaps in their paper for us to sort of look into and fill in some of the understanding. And in addition, having this experimental data from our collaborators also made a lot of sense to go after this problem. So we looked at this problem carefully, and for details, I would recommend this 2019 paper from the JCP that just came out just before the pandemic happened, in fact. So basically from our calculations, what we've noticed, in fact, is there are several timescales that are actually at play here that go from the Fe2, the excited Fe2CN6, all the way to what I call the aquated state here, in fact, where you see a water actually replacing the cyano in this case. So this reaction, in a broad sense, actually is very important for ligand exchange processes. So what we found, and these results are still preliminary because we're still working with some experimentalists, the system takes about 200 femtoseconds for, in fact, to detach one of the cyano groups. So what ends up happening is you excite onto a singlet state, and then there is a very nearby triplet that happens. So the system ends up in a triplet, and once you're in the triplet, the cyano starts to come off. And our preliminary simulations are actually tell us that this cyano group comes off at 200 femtoseconds, in which we're actually still locking down the time scale for this early process. But what was interesting as far as this 2019 study was concerned was when you've lost a CN ligand, you have this pentacoordinated system, in fact. And what we noticed from our QMM simulations was we actually found an oscillation between what you'd call a square pyramidal system and a trigonal bipyramidal system of these two kinds. And the oscillation between the two was about 3 picoseconds, in fact. And of course, you can also simulate the long time behavior, which basically gets you the aquated system. But the intriguing thing was, why does this reaction take 20 picoseconds to become fully aquated? And that was one of the big burning questions when we got into this project. Now, this dance between these two 5-fold coordinated geometries was actually a bit of a surprise to us, in fact, and this was very interesting. So we got into this system a little bit more. And what we found was for every 3 picoseconds of oscillations, the system actually stays in the square pyramidal form for about 500 femtoseconds and the trigonal bipyramid for 2.5 picoseconds. And what that tells you is it's something significant, because it's only in the square pyramidal form that the cyano group that the water can actually come and attack the cyano, the complex, and fill that spot left over by the cyano group. Whereas in the trigonal bipyramid, there isn't a space for the water to come in fact. So you see that only about 15% of the time is this geometry available for aquation, in fact. So this gives you a mechanism for why this reaction takes 20 picoseconds, in fact. So what I should point out is that these are still predictions from theory. This fluxionic type dance that we discovered in our QMM simulations is still being verified by our collaborators at Argonne, in fact. So they're actually trying to lock this down to see if there are experimental probes they can use to actually verify if our predictions are correct or not. And I'd encourage you to go and look at this Global XAS Journal Club talk given to me by my collaborator, Anne-Marie Mach, on the 2nd of June of 2020, where she talks about the experimental side of this study as well. So if you can compute, like I was saying, because we can also compute these KH-XANES for every one of these species that we've got. And as I showed you earlier, this is the reference species, which is the Fe2CN6 ground state species, and the theory does pretty well. You get one feature, like I showed you, because you just got excitations into the EG. And likewise, when you go to the science, the pentacoordinated species, where I've drawn these orbital diagrams here, you can actually see that the experimental spectrum is well reproduced by the theory, in fact, in pretty good agreement. And the reason why you actually see a higher intensity here compared to the reference species is because there's actually a dipole mixing that's happening here, in fact. And likewise, if you can go into the aquated species at 20 picoseconds, where you can actually just simulate the water coming in and replacing the cyano group, you can actually see that this is also well reproduced. So a big takeaway from this, of course, is you actually even get the chemical shifts, right? You see that in the pentacoordinated species that there is a red shifting about 2.5 electron volts compared to the model complex. And in the case of the aquated species, you see a red shifting by 1.5 electron volts. And so this was well borne out by our calculation. So, the thing before I jump to the next topic, there are, like I was saying, there are these open questions that we're trying to tease apart where this early time scale, where we're actually trying to look at ultrafast scattering to see if we can actually nail down this early time scale. And as I was pointing out, this fluxionic species here, in fact, is still to be verified by experiment. In fact, so this is still an ongoing study. And I think, our predictions, this is again a case where theory leading experiment, providing experimentalists some features to go look for in the experiment. So that's the exciting part of it, for me at least. So I'll jump to another system. And I would say that this is a much faster time scale, because the other one I was talking about 200 femtoseconds and 20 picoseconds for the aquated system. This thing happens at a much faster time scale. And it's a little bit more complicated system. And the protocol that we in fact used here is the same one that we actually showed you in the previous study, where we've looked at the transition metal complex in QMMM and in the QMMM environment. So in this specific case, we were specifically interested in this quantifying the transient excited state mixed valency of this fairly large complex for the iron ruthenium complex, where you have an ion on this side, which is essentially the same iron 2 complex that I was talking to you about earlier, but happens to have this bridging C and cyano group in between. And then, of course, it's complexed with this ruthenium amine group on this side. So this is a very well-studied complex by the Barbara group way back over 20 years ago, and people have been looking at it with different probes, and we came at it from the X-ray side. So the interesting thing here in this process is, once you put it up on the excited state, roughly about 1 electron volt, you get this MMCT transition state, where charge transfer state at 961 nanometers. Essentially, basically, it's a charge transfer from the iron to the ruthenium. And since X-rays are these X-ray exquisite probes, and ultrafast probes and element specific and all the great things that you can do with X-rays, we wanted to see, once you put this up on the excited state, can we actually use X-ray spectroscopies to tease apart this change from the Fe2 plus as the electron wanders over to the ruthenium, going from an Fe2 plus to an iron 3 plus, and ruthenium going from a 3 plus to a 2 plus. And that was essentially the question we were after. So to make this happen, we in fact did excited state optimizations because of these are fairly large complexes. So what we did was we computed clusters. We also wanted to understand how the solvent orbitals were actually playing a role in the excited state process. So we actually did all the equilibration in the QMMM environment, and then constructed fairly large clusters of these complexes, and then ran excited state optimizations using TDDFT gradients tuned on the MMCT excited state. And we basically captured snapshots. And then for every one of those snapshots, we prepared a determinant that using the maximum overlap or the constraint DFT, where you can actually make sure that you have the right orbitals that are involved, because in TDDFT, you need to have a reference orbital from which you basically can take an excitation from. So if you wanted to understand how this oxidation state changes, you need to be on this excited state reference from which you do the TDDFT calculations from. And if you follow that process, in fact, you can see that as that system travels on that excited state, remember this system on the ground state is an Fe2 plus and doesn't have this A feature that I was emphasizing earlier in my talk. So as the system starts to move over to the Fe3 plus ruthenium 2 plus state, you can actually see the emergence of this A feature slowly happening, in fact, which is telling you that the system is starting to become Fe3 plus like. But of course, it's not truly Fe3 plus like, because remember, this system is making this transition on the excited state. And these were purely calculations, of course, right? And so we had to go back to the experimentalists, and the experimentalists, of course, needed a way to understand what they were actually seeing. And they found that the best fit was about 0.6 plus or minus 0.2. In fact, in the A minus B features, the whole density corresponding to about 0.6 plus or minus 0.2. And so this A minus B metric that we have on the x-axis, which is a measure of these A minus B features, actually was a good, nice little ruler, if you want to call it, to pinpoint where what what the delocalization that was actually being tracked by the experiment. And for reference, actually, I put the star here, which is the Fe3+, which is the ground state reference complex. And we had two studies with this. The X-ray study with the transient X-ray was published in this Journal of Physical Chemistry letter paper with all the details if you want to look at how these calculations were done. And there was also an ultrafast scattering work on this as well. And this was published in Nature Chemistry, where we didn't look at any of the, we were just looking at the solvent effects on those ultrafast timescales of the back electron transfer that happens in about, 70 femtoseconds. So that was purely a molecular dynamics study. And to be specific, it was some kind of, it was actually a non-equilibrium molecular dynamics study, in fact. So more on the dynamic side of it. And this particular study was more on the X-ray spectroscopy side of it. And there was actually a global XANES Journal Club talk on this as well on August 19, 2021, that focused on the scattering side of this study. Going even farther, this was an interesting study, in fact, again, during the pandemic. And I should point out that these studies were, we took on these studies because a lot of beamlines were closed. And so we found good ways, in fact, to start moving theory forward in front of the experiment, in fact, in anticipation of the beam times coming later. So we went off and actually studied, we prepared these, we went ahead and did these theoretical studies so we could just get ahead of the experiment in a matter of speaking and be ready for them. So this particular study, we wanted to study the ultrafast signatures of excited state intramolecular proton transfer in this HPQ system. And just to give you a sense of what the system does is when you excite, when you come in with an optical probe and put the system on an S1 state, the system, in fact, makes this transition from this enol side, the proton transfers over to the enol side to the keto side, which is the nitrogen center here. For reference, on the ground state, the enol side is actually the most stable, and there's a very, very small minimum which based on the keto side on the ground state, and you can actually calculate this thing, but fluctuations would just tip the system and put them onto the enol side. So, but on the excited state, the story is a little different. So the system makes this transition from the enol to the keto, and it, of course, lives on the keto star. I would say the enol star and the keto star. In fact, it lives on the keto star for about 200 picoseconds, but it makes this transition on an extremely fast time scale. So again, we wanted to, in the theme of these transient X-ray spectroscopies, we wanted to understand if we could actually use theory to delve into the details of this system. So the first thing you do is, of course, you can do some molecular dynamic simulations on the S1 state, and our theoretical calculations pretty much nails down what the experiment observes, about 13, 14 femtoseconds. And then, of course, just like I was showing you in the previous examples, we also computed the nitrogen K-edge and the oxygen K-edge and monitored this edge as the system made this transition from the enol to the keto side, going from 0 femtoseconds onwards, and the system is already transitioned over to the keto side at 14 femtoseconds. So we monitored the nitrogen K-edge and the oxygen K-edge, and you can actually see the emergence of new features, in fact, which is a result of the transient nature of these X-ray spectra. But what was interesting, in fact, because what we thought would we do is we ran these simulations over multiple trajectories. And this plot here is essentially a stack trace of 20 trajectories, in fact, where we monitor the A, B, and C peaks features in the nitrogen K-edge, in fact. And then this particular, we drew a line, we basically took a cut across the B feature and plotted it here. And we did the same thing for the oxygen K-edge, where we monitored the A feature. What was interesting, of course, was when we saw these oscillations, the student that I was working with, Caroline Lowe, basically said, okay, we know we could always Fourier transform these oscillations to see if they link up to some modes that are actually present in the molecule. And what we found was, of course, these modes were, in fact, correlated with the backbone of how this molecule was actually vibrating on the excited state. which was actually quite beautiful. In fact, you can see how these vibrations, in fact, modulate the X-ray spectrum, in fact. So that was actually a very, very nice result that we found in this paper. And we're actually looking forward to doing some experiments on this system going forward, because with these predictions in hand. And what I should point out is that one obvious question that can come up is, of course, we're moving a proton in the system, right? And a proton, if you want to treat the system precisely, you need to bring in nuclear quantum effects. In this specific case, we didn't do the nuclear quantum effects because this whole system was downhill. And of course, if you happen to have a barrier on the excited state, you need to bring that, you need to bring the nuclear quantum effect into consideration here. In fact, and that is something we're actually currently looking into. And for additional details of this paper, I encourage you to go and look at this publication from 2021. And now transitioning over to another illustration of what we've done recently. This came out just recently, just maybe less than a month ago. We were interested in looking at, X-ray and optical circular dichroism, in fact. And the one thing we were interested in seeing is, can we illustrate the local and global nature of these two probes? And our postdoc, Victor RIXS, discovered this molecule, the 12-helicene racemization, where 12 actually stands for the number of benzene rings in the system. And what he found was this work by Barroso and co-workers, which basically had chalked out a pathway going from the transition state all the way down to the chiral state here. And the transition state can be imagined as a racemic state, because you could think of the molecule actually having two opposing sides to it. And so there's got no chiral signal, in fact, here. Whereas if you basically come down the hill down here, all the way down to the chiral state, you start becoming some chirality, and this little moving on the side kind of shows you those two geometries. What I don't plot here is, of course, is the time scale. The time scale here is much, much slower than what I've been showing you so far. This is actually over a time scale of several hundred picoseconds, in fact. So what we noticed was, starting the geometry, what we did was we started the dynamics from the transition state, which is a racemic state, all the way down to the chiral, and over this 200 picosecond time frame. And for this, of course, we combined, we ran these simulations with this NextMD code, which is another open source code developed out of Los Alamos, using a semi-empirical Hamiltonian that we had actually tested for the system to make sure that the dynamics was good. And once we did that, at 300 Kelvin, we snapshotted a large number of these geometries along the pathway and computed the X-ray circular dichroism and the optical circular dichroism signals with NWChem with these settings written right here, in fact. But eventually what you can, so the net result of this paper, in fact, is quite interesting. So if you start from the transition state here, you see that the optical circular dichroism, because it's a global probe, kind of gives you a sense that it's completely racemic, as we expected. But if you tag, these little these tags here, in fact, you see these two atoms here that replace the hydrogens with two different colors are the fluorine and the chlorine centers. And you notice that if you monitor the X-ray circular dichroism signal at the fluorine K edge as well as the chlorine K edge, you notice that you actually start to see chirality even at the transition state, because these atoms are X-rays, again, illustrating the local nature of this, basically probes the local chirality of the system, in fact. So even with all these fluctuations that we had, despite these fluctuations, you actually can see that the X-ray circular dichroism signal does pinpoint the local chirality in the signal. So this might be obvious to people that work in circular dichroism, but I thought we thought that this was a very, very nice illustrative example where one can use X-ray circular dichroism as a local probe of chiral changes in a molecule. So the ongoing work, summarizing the first part of this talk, essentially, we're still looking at the photoacquisition of the early time that I was telling you about. We're looking at the scattering signals. We're trying to disentangle this fluxionic species. And to go further from this iron-ruthenium complex that I was telling you about, we are actually looking at a trimer, an iron-ruthenium trimer. But in this specific case, we have a 3+, 2+, and a 3+ here. And there's a metal-to-metal charge transfer that takes place from going from the iron to the ruthenium, or it could actually involve both the ions. So these are studies that are ongoing. And for the proton transfer type systems, we're actually including nuclear quantum effects, and hopefully we'll have some capability that works with NWChem at some point, and we'll release a protocol that people can use. So I'll transition to the next part, which is valence-to-core, or the reverse process, where we're actually looking at de-excitations from the valence to the core in this specific case. And the commonly used acronym is V2C-XES. And because there are XES can have different branches to it, but this is a very specific branch of the X-ray emission spectrum. So for people that are uninitiated again here, the X-ray emission lines, in fact, can be broadly categorized. And you see the alpha, which are essentially the 2p to 1s transitions. And then you've got the 3p to 1s transitions, or the k beta lines. And then you've got these little satellite lines that come from the valence. Now, what is interesting is, of course, these are the most intense, and then followed by the k beta lines, and then further less intense are these satellites, which actually have a lot of very good chemical information imprinted in them, in fact. And this cartoon here at the bottom kind of tells you what this V2C-XES that I'm going to talk to you about basically summarizes you. So you basically create a core hole, you ionize the system, and then you just see the system just start to de-excite down to the 1s state. So there is actually lab-based experiments pioneered by Jerry Seidler's group at University of Washington that I've been collaborating with recently that basically produce these signals in exquisite ways. So that is something that you might want to keep a lookout on for those publications as well. So the computational approach that we have in NWChem is similar to some other implementations that are out there in the quantum chemistry arena. But we rely within the TDDFT framework. And so what we do here is we set up a core hole, and we make sure that, people working in quantum chemistry have to know that you must make sure that you don't collapse into this core hole. So you have to maintain this core hole while you do the SCF. And once you have the SCF with this core hole present, you pretty much use that reference, like I was saying, that core hole reference as your reference state to perform response calculations, in fact. And so it's basically what you naturally see are the de-excitations into that core hole reference that basically give you the emission spectrum. What is interesting about this is in TDDFT, even if you're working within the single excitation framework or de-excitation framework, you would have noticed that each excitation is essentially a sum of single slated determinants, so several single slated determinants with different amplitudes. And so you actually get this multi-determinantal character of the excited state. So you're going beyond sort of a cone-sham look into this picture, because you can still do these calculations sort of with cone-sham orbitals for the initial and final states, and then compute the sandwiches that would require you to get you the oscillator strengths. But doing it within the TDDFT framework naturally gets you this multi-determinantal look into these excited states, just like you have for the absorption side. And this is an implementation we published almost 8 years ago, in fact, where we illustrated this work. And in fact, in that paper, we looked at the 3D to 1s valence-to-core X-ray emission, where there's a lot of, I would say in the literature, there's actually a lot of examples of 3D to 1s literature examples, in fact, because this is a very, very good probe to understand what the valence orbitals look like, right? So if you combine the, say, iron KH XANES with the iron KH valence-to-core X-ray emission, with those two, in fact, you get a good look. You'll actually get a good look of the whole bonding of a molecular complex. And as you can see, you get different features that are enhanced depending on the, these are actually a whole host of 3D transition metal complexes, model complexes that we use. And more recently, in fact, 2 years ago, we looked at the 4D to 2P transitions for the row below it, in fact. So there are not very many beamlines that do this. And we had access to some very nice data from SSRL. And that allowed us to test our implementation against experimental measurements. But the take-home message for the students in the audience is the valence-to-core X-ray emission, the intensity of the peaks are directly proportional, basically proportional to the contribution of the transition metal 3D and 4D orbital characters of transition. That's a very big take-home message of this. And the main peak, right, in this case is dominated by the 3D to 1s, and in this specific case, 4D to 2p. And this encodes a lot of information about the effective charge and the ligand field strength. So as I was telling you just a few minutes ago, if you combine this with the XANES, you actually get a very good look of the local chemistry of the system and the bonding environment. So just to have a quick call out to work that I did with Jerry, I'm not getting into the details of this. We, in fact, looked at the sulfur 3p to 1s. In the case of sulfur, in fact, 3p to 1s, which is the valence-to-core, it's essentially is also the K beta. And because we had shown success with our theories, Jerry suggested, why don't we just do a whole host of them and see, basically go with theory and see if we can actually do some machine learning. And I know Tom's been working on it very actively over the last several years. So I thought I'd just call this out, where we in fact looked at a whole host of sulfur complexes in this paper here, and a whole host of phosphorus complex in this paper here. And Jerry's students, Samantha Tetef, in fact, did a machine learning study of this to look at the patterns that form over different oxidation states and bonding environments. So for the machine learning interest in this audience, I would recommend looking at these two papers. In fact, I'm not a machine learning expert by any stretch. My contribution to this work was purely from the X-ray spectroscopy and implementation side of it. But I think with the wave of machine learning and X-ray spectroscopies and work that Tom's pioneered, I think this is actually a very, very topical area that is of interest. And you can do even battery materials, right? You know, this was again an illustrative study where we looked at a solid state sample where we carved up these clusters, which is not a periodic way of doing things, but if you work with a molecular code, you have to come up with these clusters that you have to construct. And the theory actually does reasonably well. It brings out the splittings. And of course, the biggest issue here is, of course, constructing these clusters, which is not a trivial matter. And in fact, the system gets more complicated. And so we were looking at different oxidation states of vanadium here. In fact, just to sort of show you a different example from the molecular system that I was showing you earlier. So this is, again, this thing just as a prediction, because this is something we've actually thrown out to our experimental collaborators, bringing it back to the photo equation that I was showing you. We computed the valence-to-core for these two species again, these pentacoordinated species that I was telling you that are still to be nailed down. But, you know, if you look at the valence-to-core X-ray emission, they pretty much lie on top of each other, except for the small little difference here, in fact. Although there is this little chemical shift, and I hope our experimental collaborators can actually tease that out, I think it'll be difficult because, as I was telling you, for every 3 picoseconds, the system only lives 15% of the time here and 85% of the time here. So if you get something from the experiment, you'd have to appropriately weight it. So it might get washed out. So, but I'm still hopeful. So this is, again, this is a prediction that we've reached out to our experimental collaborators with. So in the last part of the talk, so I've shown you absorption, I've shown you emission, and now there is a technique that's actually become quite popular in the molecular side of chemistry. This probe has been very, very, it's a very well-established probe in condensed matter physics, resonant inelastic X-ray extraction, which is essentially an X-ray Raman process, right? If you think about a standard Raman process where the UV light rides on top of the vibronic spectrum, if you translate that whole thing over to the X-ray side of it, the high wave, the high frequency, that's the analog of the UV in standard Raman becomes the X-ray, and it's riding the UV excitations, in fact, like the vibronic in standard Raman. So Raman, resonant inelastic Raman scattering is actually a really, really interesting probe that allows you to connect up and correlate the valence and the core excitations. And in a snapshot, basically what you do here is I was showing you this valence-to-core, which was non-resonant, where you basically excite the core level all the way out. In this specific case, you do something different. So you basically excite a core electron to an unoccupied target, unoccupied state, and then you basically look at all the de-excitations from that state into other levels, in fact, in the system, as the system starts to de-excite. So there's two branches to this, right? You can get the elastic branch where what goes up comes back to the same place, or you can get an energy transfer that happens, in fact. So in a nutshell, this is basically what the RIXS spectrum encapsulates. So from a formalism standpoint, because we are working in this TDDFT framework, right? So we wanted to see how we would extend TDDFT to do this kind of thing. And the object you need to compute for the RIXS is essentially this Kramers-Heisenberg equation, where you get these two matrix elements, this matrix element being the excitations out of the core, and these connected to the matrix elements are the de-excitations to the final states. And you need a mechanism to compute these matrix elements. Now, if you go into formal response theory, the way to do this is through quadratic response, with the excited state transition moments come out to be the double residues of the quadratic response function. But there's, in fact, classic papers going all the way back to 1982 by Dalgaard, in fact, which showed that, the quadratic response, in fact, you do get these things called spurious poles when the transition energy between two excited states matches the excitation energy between the ground and another state. So in fact, you get this extra pole in the denominator if you actually go and inspect these equations. And you can get these singularities present there, in fact. And so there are what I would call landmines in the theory. And it was basically a beautiful paper written by Parker and Philip Furka, in fact, in 2016, that analyzed this in detail over a broad class of theories, in fact. So given that as the backdrop, and in addition, quadratic response is also very numerically intensive. And we didn't have an implementation in our TDDFT code in NWChem to take advantage of quadratic response. So we said, can we do this sort of via a linear response approach where we've got all these pieces that allow us to put this equation together? So we relied on, we basically appealed to this idea called the pseudo wave function approach, because when you solve the Casita equations in TDDFT, you get these solutions, which are essentially CIS-like solutions. And you can interpret them as reasonable wave functions, what we call pseudo wave functions, and many other authors have also referred to them as pseudo wave functions. And if you treat them as pseudo wave functions, you can actually set up working equations to compute these matrix elements. And that's literally what we did here, in fact. And the details are in this JCTC paper that came out in 2021. And with that, how does it do, right? And so one thing you can ask is, what am I missing here? So the key point that's missing in composing these matrix elements using these pseudo wave functions is essentially you're basically neglecting the second order polarization, density relaxation in quadratic response theory. So what I'd like to call this specific approximation is like a static approximation that's neglecting this density relaxation that's there, that comes from the true quadratic response. Now, how does this do? So before we get into the predictions of the 2P 3D RIXS, remember that when you do the RIXS, you need to have an absorption side and you have an emission side, right? So when we want to do 2P 4D RIXS, we need to see how our 2P to 4D transitions do first. If our initial core excited states don't do well, then I think then you would say, well, the RIXS spectrum is going to come out all wrong. So this is sort of a baseline number we did, a baseline calculation we did here. Again, going back to our experimental colleagues at SSRL who gave us this data. And this is the X-ray absorption partial fluorescence yield, as well as the HERF-D for a set of model complexes. And these are our calculations on the side here. And you can see that the calculations actually work pretty well. And these are at the L3 edge of ruthenium. And some of the details here are listed. So of course, we have to include relativistic effects because ruthenium is in the second row. And you have to make sure that you have the proper co-contractions. And we've done this at the Zora level. The one thing I'll point out that we haven't done in this case is because the L2, L3 splitting in ruthenium, the spin orbit splitting is about 100 electron volts. we basically, for practical reasons, we just ignored the L2 and just looked at the L3 manifold, in fact. And if you look at the 4D spin-orbit, it's roughly about 0.1 electron volts, which is well within the broadening that we used in the spectrum. So that is an approximation to this. And I'm not saying that you can completely neglect the spin-orbit, because spin-orbit also factors in some rules that one has to pay attention to. But for the purpose of this calculation, that approach was sufficiently accurate. So given that the absorption part was reasonably brought out by our theory, we decided to go and do the 2p40 RIXS in 3 model complexes again here. And this map here is a standard look on what a RIXS map looks like. You've got the absorption axis and you've got the emission, the energy transfer axis, as I was telling you, because these are inelastic processes. And you can actually take cuts along these various lines and produce spectrum, 2D spectrum like this. In fact, where you have the intensity versus the energy transfer. And the solid lines are the experiment, and the other 2 lines are, in fact, calculations taken across 2 different cuts here. And you can see that the calculation does reasonably well. There is some differences here. The elastic line, of course, is overemphasized in the theory in this case, but that's not the region that we're mostly interested in. But so the theory, what I would say, it does reasonably well. And it allows us to get away from doing full quadratic response to treatment. The one thing I should point out here is, of course, you have the core excited states, and you've got the core excited states, and you've got the emission. So, we had to couple, say, order 100 core excited states, and of the order of 1,000 valence states. In fact, for one of these complexes, I don't remember, I think it was for this one. So that's actually a very prohibitive calculation that you have to sum over the intermediates, which would become extremely prohibitive for a quadratic response treatment, in fact. So I think so what I would call the sufficiently accurate functional treatment actually gives us access to the 2p40 RIXS for these interesting complexes. And we're actually using this for other systems that we're actually interested in looking at. So with that, all of these, I'd like to acknowledge a broad range of collaborators. And in fact, as a theoretician in X-ray spectroscopies, it's always great to have wonderful experimental collaborators that can work with you and share data with you that you can shed light on. And I'd like to acknowledge postdocs that have worked with me, Daniel Nascimento, who's now at the University of Memphis, Shal Mukamal and his team and his postdoc. I've had a long, very nice collaboration with Munira Khalil at the University of Washington, Department of Chemistry. And the names that I've underlined are the names whose work that I actually illustrated here in this talk. Jerry Seidler and his group, people at SLAC, Elisa Biesin, who work closely with me on the Ultrafast iron ruthenium complex scattering, now is a staff member at PNNL. And Anne-Marie March, my collaborator at Argonne, that basically introduced us to this photoactivation system. And with that, I'll stop and I'll take questions.
Okay, brilliant. Thanks very much for a really, really great and interesting talk. So as always, if you feel free to write a question in the chat, or alternatively, you put your hand up and then I can say, can I mute? I'll just